Biochemical information exists at multiple levels of granularity. In coarse form, data about proteins and RNAs exist in various public databases on the internet. However, most of the info you want will require that you read papers. One of the reasons E. coli is so popular in genetic engineering is that everyone else is using E. coli. It has simply become a field standard prokaryote, just as yeast is a standard simple eukaryote, and various human cell lines are standard models of the human system. Because others work with E. coli, there is a lot of information about people putting genes into this organism. E. coli has some near phylogenetic neighbors like Citrobacter, Shigella, and Salmonella. These organisms are almost indistinguishable from E. coli. These strains are as similar to your lab strain of E. coli as are the pathogenic strains of E. coli are to one another. Additional members of the Enterobacteria family include Yersinia and Serratia, and these are also very similar to E. coli in terms of the housekeeping genes, so the ribosomes, replication factors, and primary metabolic enzymes, and so forth. Because of the similarity of these organisms, any information about the function of a gene is likely to carry over to your lab E. coli. This includes properties like gene expression and regulation of promoters, at least from a qualitative perspective. By convention, prokaryotic genes are written in italics with the first letter lowercase and all letters italicized. Proteins encoded by those genes are written with the first letter capitalized and no italics. Not all genes will express in all cells. Most prokaryotic coding sequences will express in E. coli, unless they are toxic, if the subcellular localization is preserved. 50% of yeast or archaeal proteins will express in E. coli as long as they aren't over 1,200 amino acids in length or so. If a gene fails to express, screening several, like 4 homologs of a yeast or archaeal coding sequence, often will identify ones that can express in E. coli. It is a property of a gene decoupled from its molecular function. It's about folding or stability as to whether something will express in a cell or not. Mammalian coding sequences, particularly for extracellular proteins, have a much higher fail rate, and they will often make inclusion bodies, which are insoluble aggregates of the protein. Genes synthesizing a eukaryotic coding sequence with E. coli codon usage will often improve its ability to express, but not always. The main culprit for poor expression is often the use of a rare arginine codon such as AGA or AGG, which is common in eukaryotic and archaeal genes. Proper disulfide bonding is often a problem with proteins who are natively extracellular mammalian proteins. You'll often find that proteins express or not express in classes rather than as individuals. So evidence that one protein does or doesn't express is often evidence that a related protein will or will not express. For example, cytochrome P450 enzymes and polyketide megasynthases are infamous for not expressing well in E. coli as a class. Though a lot of reading must be done today to find out whether a gene can express, the ability to synthesize many genes and ask questions about their expression in multiple hosts now exists. There will likely be databases that house such information in the next 2 years. However, for now you must mine the literature for observations about expression or test it yourself. The best evidence for expression is a direct observation. If you do queries such as coli expression XYZ, where XYZ is a gene name, you might find some papers. A popular journal title from the 80s and 90s was Cloning and Expression of XYZ, and in those studies they did exactly what you were looking for. Crystallography papers are a great source for this type of data since they had to produce and purify large amounts of material to do their studies. Just read the methods and materials section of papers about a given protein and look for the host organism. E. coli is the most convenient organism for protein purification, so if they did not use E. coli, there is a good chance that they tried that first and it didn't work. However, if they were able to purify protein from E. coli, it must express just fine. One caveat to that inference is that sometimes people will overproduce a protein in E. coli and it will accumulate, but it will be insoluble. The buzzwords here are inclusion bodies and refolding, which means they didn't actually express soluble protein. They purified the aggregates and then dissolved them and refolded them in vitro. The best way to find papers of this sort is to do a PubMed structure search for the protein name, then find the materials and methods section of the paper associated with that structure. Often you can find papers about crystallization of a protein by searching PubMed for crystal XYZ or structure XYZ-like queries. The best way to identify information about transcriptional elements is searching for assay methods. Some of the buzzword experiments associated with gene expression are microarrays, RNA-seq, LAC-Z assays, and so forth. So you might search for microarray, coli, radiation to pull out papers that do some sort of microarray analysis on E. coli with and without radiation. Your ideal situation is a paper in which the authors placed GFP behind a promoter on a plasmid and then measured fluorescence under various conditions. However, you won't find that much of this experiment in the literature. Most molecular biologists like to study gene expression in the genome to preserve the copy number in context to avoid artifacts. For such an experiment, LAC-Z is more popular than GFP because it is detectable at lower expression levels than is GFP. Searching PubMed for things like VIR-F, LAC-Z, coli will most likely pull up papers that study VIR-F expression in E. coli using LAC-Z assays. Google Scholar or vanilla Google searches can be handy for these sorts of things. For example, authors might use LAC-Z in their experiment, but in the abstract not mention it, or perhaps we'll call it a Miller assay or a galactosidase assay. Doing a full-text query for ONPG, which stands for orthonitrophenyl galactoside, the substrate used in such an assay, can sometimes pull up papers that you don't get with LAC-Z as the search token. If you are searching for things by a specific method, your queries would look like XYZ plus GFP or LAC-Z, PhoA, alkylate phosphatase, microarray, northern, or western. If you want to use words associated with an experiment, that you might try Miller or ONPG. In all these cases thus far, I am dealing with situations in which you know the name of the gene you care about and want to find out about its regulation. This is the easier direction, and some of this data is already cataloged in places like RegulonDB. The other direction is more challenging. Here you have a condition and you want to know which genes are affected by it. For example, let's say you want to find promoters in E. coli that are activated during anaerobic growth. Here you'd replace the gene name with the condition name, so anaerobic coli Miller would pull up the sort of things you want. If what you care about is more abstract, things get challenging. Suppose you want to find any combination of transcription factor and a promoter it activates. You can try searching for promoter transcription factor activation and you'll pull up all sorts of interesting things, but there's currently no way of efficiently performing this type of query to find what you want.